we like biodiversity in our plants, we should also really like biodiversity in our fungi, because just like plants, fungi can fill different niches in the soil. They have different plants they like to associate with or not, different micronutrients that they specialize in. And so finding an inoculant that's biodiverse you'll have the best effects.
Hello, everyone, and welcome back to Curious by Nature with Deep Roots. I'm your host, Sydney Ross, and we have a great show for you today as we dig into terrestrial fungi known as Arbuscular Mycorrhizal Fungus. We'll learn how they support soil health in the context of prairie reconstructions and their effects on native plant species. It's weird, it's cool, and boy howdy, we're in for a treat today. But before we get started, I'd like to make a few announcements. First, I'd like to thank Johnson County Park and Recreation District and the Missouri Department of Conservation for their support of Curious by Nature. Both organizations do invaluable work to protect nature in Kansas and Missouri so our community can experience and appreciate it. I'd also like to thank our viewers and donors like you for making programs like this possible. If you're watching this on YouTube or listening to the podcast, please comment, like, and subscribe to our show so more nature nerds like yourself can find us. And if you're able, consider making a donation. Our next episode, DIY Simple Stonework with Kyle Johnson of Burrock Ventures, airs on Thursday, August 20th at 12 p.m. Central. Kyle will take us through the process of doing our own hardscaping at home. This is something I'm especially interested in because I'm like, sure, I can put a rock in a spot, but I'd love to know how to do more advanced things like creating small stone walls, dry creek beds, flagstone footpaths, and more. So if you're like me and want some practical advice for ways to create cohesion and accessibility in your garden, you won't want to miss this episode. For our live audience, you need no reminder that the live broadcast is a great way to ask your questions to the experts. So be sure to tune in on August 20th at 12 p.m. Central for this exciting presentation. It's First Friday, or nearly, and you know what that means. It's time for our monthly Native Landscape Chat at the Anita B. Gorman Conservation Discovery Center. So join me tomorrow in person from 1 to 2 p.m. as we walk through the gardens at the Discovery Center, see what native plants are blooming, and see some cool faunal connections. It's also a great opportunity for you to bring your gardening questions, and we can dive right into it right there in the landscape. This is a free program, but be sure to register in advance as spots are limited. We have another exciting in-person opportunity, a guided naturalist experience coming up on August 27th from 6 to 7:30 p.m. at Ernie Miller Park. And Deep Roots' very own Kay Thomason will be leading this walk geared towards community science. So take a stroll through the woods and prairies while learning how to use tools like iNaturalist and Merlin to deepen your understanding of nature. If you've ever gardened with Kay, you know she's full of knowledge and a ton of fun. Plus, this is a great way to learn how your impact as a community supports conservation efforts right here at home and beyond. Be sure to register for this exciting program. As always, Curious by Nature is recorded, and all the resources, including registration links for events, will be sent to our live attendees following the show. Questions you have today can be put in the Q&A section and will be answered at the end of the show with our guest experts. If you've tuned in before, you know we always do a pop quiz at the end of the show, and y'all, we have some exciting prizes for today. So there will be one winner. So make sure you're paying attention. Let me make sure you guys can see this. I have two, here we go, maybe. Two Shawnee Mission Park and Big Bull Creek posters from Johnson County Parks and Recreation District. These are stunning. Let me see if I can get a better shot of it. There you go. You can see there's a meadowlark here and some oak trees. And Shawnee Mission Park, you can see, I'll just describe it to you. You can see the shelter, you can see the view of the lake. They're really stunning, high quality prints. I have one of Kill Creek Park. So make sure you're paying attention today and stick around for our live Q&A, which will be at the end of today's program. All right, Let's go ahead and bring out today's guest experts and welcome Dr. Liz Koziol and Matt Garrett to the virtual stage. Dr. Liz Koziol is an assistant research professor at the University of Kansas, associate curator of the International Collection of Vesicular Arbuscular Mycorrhizal Fungi, and founder of MycoBloom LLC. Her research focuses on the role of native mycorrhizal fungi in restoring grasslands, improving soil health, and increasing biodiversity. Dr. Koziol's work bridges conservation, restoration ecology, and agriculture with an emphasis on developing science-based fungal inoculants that help build resilient ecosystems. She collaborates with researchers, land managers, farmers, and conservation organizations to translate soil microbial science into practical restoration and management solutions. And if you attended our annual Planet Native conference this year, you saw Dr. Koziol in our exhibition hall with microscope in hand, checking out examples of arbuscular mycorrhizal fungi. So cool. And then I'd also like to welcome back Matt Garrett. He is the Natural Resource Manager for the Gold Medal award-winning Johnson County Park and Recreation District. He's dedicated his entire career to environmental education and natural resource management in the Kansas City region. Matt is a regional conservation leader focused on large-scale prairie restoration, invasive plant removal, and conservation planning. In his spare time, he mountain bikes and takes many, many walks with his two dogs, Anna and Birdie. Welcome back, y'all. It's so great to see you. Let me see here. I'm gonna get Liz up on here. Oh, here we go. I can hear you, but I'm gonna have you start your video. Perfection. Welcome. How are you both doing today?
Glad to be here. Excited for another program and glad to have Liz with us today because I am not a specialist in this area.
Fantastic. Well, that's exactly why we wanted to welcome you both here. So, thanks for that, Matt and Liz, Dr. Koziol. We're thrilled. We had such a great time working with you out in the fields a few weeks ago, and we're excited to dive in a little deeper today in the world of fungi.
I'm excited too, and thanks everyone for being here.
Absolutely. So before we get started, I just want to see Dr. Liz Koziol, what are some cool projects or events that you're working on currently?
Well, this summer I have two different things I'm celebrating. First of all, it's my 10-year anniversary at the Kansas Biological Survey, which is at the University of Kansas and where I do my research on native habitats and sustainable ag and, of course, fungi. So yeah, 10 years on that. I'm pretty excited. And then last month, celebrating another 10-year anniversary. It was the 10 years since I started my small business, MycoBloom, in which I provide native fungi for people to use in their own landscape projects.
Congratulations on celebrating your anniversary and continuing your awesome research and business, getting the fungi out into the world. Who knew that you knew that we needed this, but I think many of the folks in our audience will be surprised to learn just how important our fungus is in all types of ecosystems. So how about you, Matt? What's going on with Johnson County Parks? Got anything exciting happening right now?
Yeah, we're in the middle of the growing season, and so it's either 110 or it's raining. So getting natural resource management tasks done has been really interesting this summer because the conditions have not been awesome for a lot of the work we do. So we're getting new staff up to speed and just kind of rolling with what the weather brings as we kind of get towards the end of the growing season and start planning some of the new prairies we're going to work on and some of the honeysuckle removal projects, and like we're prepping for that winter work. So we're just kind of in the real heavy end of the growing season and getting ready to pivot to a lot of the habitat restoration projects.
Very nice. It is one of those things where it's like, wow, the conditions outdoors make it kind of difficult to get things done, but we persist because we understand this is just the way things work. So,
Yep, it's important work, but everybody's looking a little gaunt and a little tan right now.
no doubt, and maybe a little itchy too.
Yep.
All right. Well, we're going to go ahead and jump into today's episode. I hope you all enjoy a fungus-fueled prairie health as we explore arbuscular mycorrhizal fungus with Dr. Liz Koziol at Johnson County Parks and Recreation District. Enjoy.
Hello, I'm Liz Koziol, and I'm a research professor at the Kansas Biological Survey, and I research rewilding fungi in prairie restoration. And today we are at Shawnee Mission Park, Pump House, Prairie, looking at our second year restoration experiment. So Arbuscular Mycorrhizal Fungi are a below-ground soil microbe, and they're really good at collecting nutrients from the soil like phosphorus and calcium and water. And they're symbiotic with plants, and so many prairie species associate with these fungi, and they rely on the fungi to associate with their roots, and then they have this hyphae that branches all throughout the soil, and that hyphae collects the nutrients and brings it back to its plant host. And in exchange, the plants feed the fungi carbon, the sugar that's required for those fungi to grow. So both partners are getting something from this symbiotic relationship. So my research in Kansas first started about 10 years ago when I moved here for a postdoc position at the Biological Survey, researching perennial crops and perennial restorations. And this site here at Shawnee Mission Park is in its second year of restoration.
So I'm familiar with fungus, right? But I don't quite understand AMF or Arbuscular Mycorrhizal Fungi. So you explained like why it's important for our prairie health, but how do you get it into a prairie?
Great question.
And what are the effective ways? What are the ineffective ways of doing that?
So the thing that makes our research program unique with regards to mycorrhizal fungi is we're working with old growth fungi that we isolate from remnant prairies. And so we'll go to a prairie that's never been plowed and take a small amount of soil and collect the fungi from that soil. And then after that, it's a ton of microscope work. And so I'm in the microscope looking at these teeny tiny single cells and sorting them by color, shape, and size. That's generally how I tell the species apart. And then I kind of grow it up in the lab in a media with the plant hosts because they're obligately dependent on plants. And after that, we have the stuff. It's like a granular mix that has some roots with fungi in it, some free-floating spores, and then we put that out into restoration. And so we've done restoration experiments with these old growth fungi, a ton of states across the U.S. prairie, and usually we're working in small, like 1 or 2 or 3 meter square plots. And so the benefit of partnering with Johnson County here and working in these sites is we can test application techniques at scale using things that land managers might use. So oftentimes for our research experiments, we're tilling it into the soil or maybe putting it on the roots of little nurse plants that we're transplanting into restoration. But obviously things like transplanting seedlings you can't really do even on a restoration of this size, like 3 acres, let alone one that's 15 acres or 100 acres. And so really trying to think about how can we put this fungi out into the world was the goal of these research questions.
And so I've seen like you can buy Arbuscular Mycorrhizal Fungi online. Like you can just buy granulars. Obviously I don't have acreage like this myself, but I have a little plot at my house of a little native garden. Is that effective to like broadcast granulars?
I think it depends on the specific inoculant that you're buying. I think granular ones are the best. So these fungi are living things, and so they don't necessarily want to be like in a liquid form for a long time. So I'd probably stay away from any sort of liquid concentrate. Granulars tend to be the best in terms of maintaining spore viability. And so one thing that we've done at the University of Kansas is look at a lot of these granular inoculants that you can buy on Amazon or from Ace or Menards, whatever. We went and bought a bunch of products and looked at their quality. And we found that a lot of them didn't have any mycorrhizal spores in them. So they claimed to be mycorrhizal inoculants, but there actually wasn't any in there. And many of them that had mycorrhizal spores, we grew them up with plants and found that actually they didn't grow because they were dead. They were non-viable. So that's a huge problem with the mycorrhizal inoculant industry. One thing that I've done spinning out of my scientific research is start a company called MycoBloom, and I sell granular inoculants. And my ethos with MycoBloom is biodiversity. So we like biodiversity in our plants. We should also really like biodiversity in our fungi, because just like plants, fungi can fill different niches in the soil. They have different plants they like to associate with or not, different micronutrients that they specialize in. And so finding an inoculant that's biodiverse you'll have the best effects.
I think the product came to us in, would you say, was it 30 or 40 Home Depot buckets?
Nice.
5 gal is like 55 pounds. That's my maximum weight limit.
Yeah.
Yeah.
And so we went to KU and picked up multiple buckets of the live material and then brought it on site.
Mm-hmm.
And so you mentioned that you used a different methods to incorporate it into the soil here. For this particular prairie, what time of year did you do that? And was it with the seed when you were putting seed down, or can you talk a little bit about that process?
Yeah, so it all happened at the same time. Was it March or
It was
April?
actually February.
It was a February.
I think it was like the last day of February, the first day of March.
Okay.
It was like right before a snowstorm.
Yep,
So I feel like it still counts as a winter
Yeah,
planting.
I would say it was a winter planting. I couldn't remember. We plant a lot of prairie.
Yeah.
And so we do a lot of broadcast seeding. And so we thought it would be interesting here to broadcast the seed and then broadcast the inoculant and then attempt to harrow it and get it some soil contact or get covered up in a way that it needs to be versus leaving it to die, would you say, out on the bare ground?
Yeah, so that's another thing about a lot of these inoculants. The direction that they give are things like broadcasted on the soil surface. But we know about these fungi that they're subterranean. Their whole life cycle is below ground. You know, their hyphae below ground, they make their spores below ground. They actually can die in the sun. And so that's one of the things that we're testing at the Bull Creek restoration is, can you leave it on the surface and does that work, or do you actually need to put it down below? And this experiment here at Shawnee Mission Park is using the harrow to try to rake the fungi in. So it was surface broadcast, and then the harrow kind of like drags it in to see if that would be an effective way. Because probably a harrow is one of the cheaper tools you can get to actually put the fungi below ground a little bit.
And then we're lucky this year is that a lot of times success in prairie restoration is about your year, the amount of precipitation you have in a year. And this second year has been phenomenal for this site. And so we've had just an absolute above average precipitation year, and you can see this site looks phenomenal. And so if we had planted this into 2 or 3 years of drought, we could be in a tougher situation. But for the future success, having the first 2 years of this have above average precipitation has been phenomenal. You would say in your second year of research, this site had reduced weed competition?
Yeah, we saw up to 50% improvement in native abundance in my transects, and then less non-native abundance, specifically of non-native grasses. So the most common one here is Bromus inermis, which was kind of the lawn before it was restored. And then the other big weed is Johnson grass. And so not one we like to see around too much. But you add a little inocula, you see biodiversity of natives increase, and then that kind of chokes out the weeds, we think, is what's happening.
Yeah, so the overall goal at Shawnee Mission Park is to connect all the remnant prairies with new restorations across the landscape. And so the Pump House Prairie was a cool season pasture, and it was just another area that had been taken out of the mowing rotation by the park maintenance staff, and the natural resource team realized it was a great opportunity to connect Og Prairie to the Bloomport Prairie, to the Spiderwort Prairie, to the one way, like it's just that we're stitching all of these little 5 to 6 acre prairies together across the entire park landscape. And so it was a great opportunity to work with Liz at KU and bring some long-term research to the site. So separate from just doing the research and doing the restoration work, to combine it together and see what she could learn from some of the common methods of how we plant prairie. Yeah, so I think we're seeing a lot of Coreopsis tinctoria, or like an annual Coreopsis, and we tend to use that in a lot of early restorations. It buys us some time with the public, so when things are super weedy, we get lots of questions, and so using an annual is really gorgeous in the first 2 or 3 years, and then they tend to fade out of the plantings pretty fast. And we did kind of want to mimic Og Prairie, our remnant here, so we added 5 pounds of Echinacea pallida to the site. And so I don't know if you saw it prevalent across all 6 plots.
It was in most of the transects I did.
And we have removed all the bush honeysuckle out of the entire woodland behind this.
Okay.
And so the entire 24 or 25 acres behind the prairie, we've completely opened the understory.
Amazing.
And there's a lot of new weedy prairies around here, so the turkey population in Shawnee Mission Park are pretty robust. They're not doing great across the state, but here within the park, with the removal of the invasive shrubs in the forest and then a lot of weedy, seed-heavy prairies, they've been really happy here. And long-term behind us, we have some mature eastern red cedar, and so in the wintertime, my team might begin to take down some of the eastern red cedar and begin to feather this edge a little bit more instead of having this hard cedar to prairie. we'll probably go in and begin to cut and make some brush piles and begin to open this site up and let the prairie kind of morph out into the oak hickory woodland behind it. And so it was maintained as a park, manicured landscape with rows of trees and turf grass. And so as we rewild this space, the goal would be to very slowly and carefully take down a lot of the cedar groves on the edge of the site.
So I'm going to show you how we collect the data at this prairie restoration. So I have these step-in stakes, and attached to it is a piece of string that's 3 meters long. And so I stretch this out, step it in, and then we look exactly under this string and document every species we're seeing, both native and non-native, to get a sense of richness at the site. And then along the string, I have tick marks placed every 30 centimeters. And at that location, we'll drop this stake and look at what plant species we're touching and how many times we're touching it to get a sense of the abundances of the different species. So like dropping it here, I would look down, I have a touch of a Coreopsis tinctoria, some Rudbeckia hirta, some red clover, and a Retibita. And so we do this at 10 to 12 locations along this line, depending on the transit grid that we have. And we use that to look at the abundances of the different plants that are establishing. And so yeah, this method is like pretty easy to get going. You know, if you kind of know some of your plants that are here, there tends to be the same 20 species or so. You get to know them, you can start collecting this abundance data in a way that's easy to compare across the plots. So I've sampled this last summer and then now this spring, and I hope to come back for many years to come. These fungi have two kind of forms. Their spore is sort of like their seed. It's their dormant structure and where they have their genetic information and they kind of go dormant in their spore form. But a lot of the function of these fungi is in their hyphal network. So it's like long filamentous tubes that's branching all throughout the soil. And if fungi are tilled in this form, if you till the soil and that hyphae gets disrupted, it kills that fungus. Most of these fungi can't survive if their hyphae are chopped up. And so any fungi that's kind of growing that way during tillage is kind of like removed from the system. And so if we compare frequently tilled landscapes like an ag field to an old growth prairie remnant, we see totally different fungi, different species, different composition of even the overlapping species. And so tilling has a huge effect on these mycorrhizal communities. And so if you want to maintain your mycorrhizal fungi, till as little as possible at your site. Actually assessing the fungal communities is really hard. It takes a lot of work in the microscope. We do some sort of DNA assessment sometimes too. But one of the indicators that's a little bit simpler to look at is the plant community response. So we know a lot of our late successional plants are forbs and legumes and grasses that tend to be more common in our remnants are really sensitive to these remnant mycorrhizal fungi. So we look to those plant species to see if they're establishing, to see if our fungal communities are having an effect on them. So Echinacea pallida is a plant that's pretty mycorrhizal. We see some Baptisia, some rattlesnake master, Laetris. So a lot of our plants that you would see very commonly in a remnant are establishing here at this restoration. Even though this is only year 2, we're starting to see things like Amorpha and Laetris even flowering even the second year. And then there are some plants that actually aren't very sensitive to mycorrhizae. And so we're looking at some of these right here, like Rudbeckia hirta and the Coreopsis tinctoria. Even though they're beautiful and they have all this yellow in this site, they're actually not super sensitive to mycorrhizae. And so if you have a landscape you think has a really bad fungal community, those are great species to seed in there because they'll do pretty well even if your fungal communities are not super supportive. This is my favorite prairie plant, Amorpha canescens. It's got really nice purple flowers with like orange pollen when it's in full bloom. This is a little past its flowering stage, but it's also really soft, and just look how cute these leaflets are. Oh my gosh. It is very common in remnants, but extremely rare in restoration. So whenever I see this in one of my restorations, I'm very excited because this plant's really mycorrhizal and associates with its native AMF, and it's a good sign your native AMF is working. So at what point do you think you will consider mycorrhizal fungi as a restoration tool?
Yeah, it's a great question because it's expensive. Like you and I have talked about it before, that getting the inoculant, would you say, is equal to the cost of the seed almost?
Yeah. Mm-hmm.
And so for a land manager, that's pretty big. To have a product, it's got to be really worthwhile. And so
I think if we continue to see weed suppression each year just dropping, did you say it went down 50%?
Yeah, it was 50% in the Shawnee Mission Park, and then some of our older restorations, we see that pattern continue on through year 7, 8.
So if there's an acceleration like that, and plus seeing the conservative species take hold faster, so if we start to see some of the rarer plants earlier, that would be success, and I would purchase the product and continue to collaborate and use more of it. If we begin to see things like lead plant establishing year 3, 4, 5 instead of later, I think that would be, for a land manager, it would make a lot of sense to have just really know that a prairie is going to establish and not have to question it as much and see the reduced weeds. So yeah, I think it makes sense, and so far it looks like it's working. And where we're standing here on the north end of Shawnee Mission Park, we have 50 acres coming up, and so we have more ability to continue to test it and see where it goes.
So remember how I said fungi have the two kind of forms? There's like the spore and then the hyphae. We're going to be checking out both of those.
Awesome.
And so the spore, even though they're microbes and they're single cells, they're actually quite macro. You can see them with your naked eyeball.
Really?
So like this is a slide here where I have a ton of little spores crushed, and I put a special stain on it that makes the spores turn really dark color, so you can actually see them. So that's a single cell you can see with your eyeball. So they range from like 500 micrometers, which is like half a millimeter, all the way to like just a tenth of that size. So these are like the half a millimeter size you can sort of see. And then I have some of these spores floating in water on this little microscope here, and there is a big white one and then kind of a little black one next to it. And so what we're looking at here, this is actually a fine root, so you can see the fine root, how big this spore is relative to that fine root. So for single cells, they're really large. So there's a white one here and then a teeny tiny black one there. Here, that white one we were just looking at, I put it on a black piece of paper just so you can really see the color pop. But these little white balls here, those are the big spores we can see. So yeah, when I'm starting cultures for these restorations, what I do is I go to the remnant prairie, like we visited Og Prairie, and take a soil core, and then put that soil core through, essentially it's a set of colanders that are microscopic scale. So you think like you're draining spaghetti, I'm draining spores, so the holes are really, really tiny. And then I get something like this, which is a bunch of spores from that soil, and then I basically grow them in the lab, bulk propagate them to enough to put out into scale into restoration. The fungi in their other form, which is their hyphae. It's really hard to look at hyphae in the soil because soil is dark and hyphae are tiny, but one thing we can do is look at them inside of the roots. And so what we have here is a root slide. I'll give you an example of one. Looks like this. It's just roots on a slide, and then we put a special stain on the roots that turns fungal structures really dark blue. And the plant cells kind of don't react as much, maybe a little light blue. So then if we zoom in on one of these little root fragments on this microscope here, that's what we're seeing in the screen. And this whole thing here is the plant root. And then all of this dark blue stuff is the hyphae of the fungi moving in between the plant cells. And so this is a grass root, and you can see just how heavily colonized it is with these fungi. And then these little blotchy things are arbuscules, which is how these fungi get their name. That's the site of nutrient exchange. So that's really where the fungi are dropping off their phosphorus and the things they got from the soil, and the plants are feeding the fungi the carbon that they need to grow.
Hi, my name is Kensington. I'm the natural resource intern for Johnson County Parks and Rec Department this summer. And we have musk thistle, or Carduus nutans here. It's an invasive that we spray constantly. Every plant can release thousands of seeds, and every flower head can release up to like 1,000 seeds. So they spread really rapidly, and they like to grow in like little clumps like this. And you'll commonly see them in prairies and along roadsides, along highways, by railroads. We work to apply specific herbicides to these plants to help reduce their populations and give native flowers more room to grow. the native thistle has a lighter colored underside of their leaves, while this one is all the same color. So it's almost whitish, the underside of the native plant. But yes, and as you can see also on this one, it's spread its seeds everywhere. You can see the massive amount of seeds that it releases. And that's going to be a problem. I'm going to have to spray that.
More learning to come in our live Q&A session, which starts right now.
All right. Fantastic. I'm going to go ahead and welcome back our guest experts to the stage. Hey, Liz. Hey, Matt. Welcome back.
Great episode. A lot of
I
fun.
really enjoyed it. It was so much fun. You guys you're the ones who make it fun and fantastic. So thank you for everything. And thanks to my teammate, Maddie Ball, for doing such a fantastic job producing this episode and editing it together. Thanks, Maddie. Okay, we've got a series of questions here for you all. We'll start off with Brenda's question. And folks, if you have questions for our guest experts, be sure to locate the Q&A tool probably at the bottom of your screen or maybe somewhere else on your screen and put them there rather than the chat so we can see them easily. So Brenda notes, on the microscope slide image with the grass root, was the blue in the center of the image of the mycorrhizal filaments on the root or in the root? It looked like they were inside, but that is the question.
Yeah, good question. So these AMF are called endomycorrhizal fungi, endo meaning within. So they tend to live within the root, and that's where they make those arbuscules, that nutrient exchange structure. But then from inside of the root, they're also going out and growing into the soil and collecting nutrients, so they're also in the soil. But
Dang, very cool. Well, great question, Brenda, and great answer, Liz. Okay, we've got a question here from Kristen. This one perhaps could be answered by both of you, Matt and Liz. Does herbicide application impact prairie fungi? And if so, what does it do?
Well, I'll talk about the fungi side, and Matt, you can go herbicide generally. But so one thing about these fungi is that they're obligately dependent on plants. So they can't get carbon from the environment in any way other than living inside of a plant root and the plant feeding carbon to them that it makes through photosynthesis. So if you kill all your plants, there's nothing to feed the fungi. So that's one thing. But in terms of like direct assessments of like herbicides on fungi, there hasn't been a ton of research, but some that has come out has indicated that yes, it can also reduce the viability of these fungal spores too.
Interesting. And I'm assuming, Matt, when you're doing herbicide application, it's well past the point of when you're inoculating. Is that correct?
That's correct. And a lot of times, if we're converting a field with something like glyphosate, most of, I would say, the AMF that Liz is looking for isn't present in a lot of these cool season pastures. And so the inoculation would be bringing some of that diversity back, and that they don't seem to spread really far, correct, Liz? The initial conversion and the use to get rid of the cool season grass generally wouldn't be a big effect on the native AMF.
Very good. Okay. All right. So I've got several more questions here. Let's see. I'm going to go with a question from Bridget. Bridget is asking, would applying AMF support her home native garden? If so, how does she buy it, and what's the best application method for a new and for an established garden? And I'll send that to you, Liz.
Yeah, I think inoculants can work at the home garden scale for sure. It's just fewer plants than what we're doing at Johnson County with these acre restorations. And I think it depends on the plant that you're working with. So if it's like an early successional thing, like I mentioned, that Rudbeckia hirta doesn't really need that much mycorrhizae to support itself. It has like a really great fine root architecture. So maybe if you're working with a plant like that, a little less. If you're working with something like butterfly milkweed, they're more sensitive to mycorrhizae. So it might be something you consider depending on, exactly what plants you're putting out there. In terms of where you can buy these products, pretty much everywhere these days, we see them at Ace, pretty much every home a department store sort of thing when they have their seasonal supply. It's even in some potting soils that you can buy some of these inoculants. But like I said, a lot of them maybe not super viable.
Yeah.
So that's also something to consider. Yeah.
So on that topic, I know that there are businesses out there that sell the liquid mycorrhizal fungus. If the product is fresh and applied immediately, would that be effective, or just do you generally discourage the use of liquid AMF?
I guess I haven't assessed all products, but so the ones that we have looked at that are liquid, we haven't seen any that are viable so far.
Okay. And what would be the best methods to apply granular, we'll say granular AMF into a home garden? Let's give two examples. Like, what if it's a brand new garden, and what if it's an established garden that you're just adding a few things to?
Yeah, I think it depends how you're getting your plants going in your garden. So if you're planting little plugs, which is often what people are doing in their yard, you buy some plants at the Planet Native plant sale or whatever. When you're digging your hole to put the roots down in the earth, that's where the fungi want to go. So I dig my little hole and then put, depending on the inoculant, a couple teaspoons right where the roots are going to be, and then put the plant in and bury them all together. So you want those fungi to contact the plant roots as soon as possible so that that symbiotic relationship can get started.
Sweet, that's awesome. And another related question from Val. She says, in my small garden, will the fungi ever arrive without me trying to add them?
Yeah, so Matt alluded to this a little bit. They're extremely dispersal limited. And so, their whole life cycle is below ground. They're not like mushrooms that we might collect in the road that have like spores that float in the wind, and that's how they disperse. So their main method of dispersing is actually growing from, a place they're well supported. They grow with their hyphae along in the soil, and that's a pretty slow process. And if you're growing in your neighborhood, in your yard, probably the fungal communities nearby are pretty disturbed. So the likelihood that you'll find good fungi being able to crawl slowly to your yard is pretty low. So that's an example of a time where we might want to think about amending our soils with beneficial microbes.
Awesome. Well, folks, we'll be sure to put a link to MycoBloom and some other resources in the show notes following today's presentation so you can get your hands on some good fungus for your gardens. Here's another question for you, Liz. Another one from Bridget. How sensitive are AMF and other mycorrhizal fungi to temperature changes or other environmental conditions that might shift due to climate change?
Yeah, good question. And I think that's probably what has to do with the viability of a lot of these products. They're living. So just like you and I, they don't want to get too hot, for example. And so these products, might come on shipping containers, sometimes from overseas. And so that's pretty stressful on these fungi. They don't want to be 150 degrees in a box or, wrapped in black plastic in the parking lot. And so I try to tell people they don't need to be like stored in the fridge or anything, but like room temperature. They can get as hot as up to like 120, no problem. But yeah, hotter than that, just like a human, they don't want to be, desiccated to death, dried out in the heat. And so that's something to think about. What was the second part of that question?
It's bleh. Or other environmental conditions that might shift due to climate change. So we're seeing lots of excess rain, for example.
Mm-hmm. Yeah, they can handle the rain pretty well. They don't live in totally inundated areas. So like if you're planting an area that, maybe is a rain garden now, but is going to be a pond, they're not going to live, under that water for the most part. But even places that like a rain garden that are temporarily flooded, they persist there. Mm-hmm. And in terms of climate change, actually fungi are going to love it. They're fed off of carbon. So as we see CO2 go up, plants are taking more and they're feeding more to their fungi. So yeah, they're going to be super happy about it.
Fungus rules the world. We already know this, but
we do hope this will make our prairies more resilient to climate change. And so when Liz is collecting this local ecotype AMF, and then we're broadcasting, we're getting it into the soil, we're hoping it'll make some of these plants that we're putting in the ground and the seeds we're putting in the ground, making them more resilient, having a little bit more structure to allow them to thrive early on in an early restoration. So it'll be really interesting to watch these two plantings.
that is interesting. Okay. I have a question from Jane Rose. Do mycorrhizal communities differ greatly between regions or in Kansas prairies versus Missouri prairies? And then, yeah, so that's my question, I guess, for you, Liz, and I have a follow-up question for you, Matt.
Yeah, good question. I would say we've done a lot of research comparing like at a location how different they are in disturbed and not disturbed environments. And we've done some assessments like Kansas versus Indiana, how different are they? And yes, they are different fungi there, and there's different abundances of even ones that are similarly overlapping. But what we see time and time again as we've done these research experiments now, my lab's been doing them for about 20 years, is that the old growth fungi that are supposed to be at the site, the endemic ones at the untilled prairies that we have, like our reference communities, those are super beneficial fungi. And so that is one thing that's really consistent. They're more beneficial than what we see in disturbed landscapes.
Interesting. And Matt, could you speak to the differences between prairies that are commonly found on the Kansas side versus like maybe some of the ones we saw out in Gray Summit or just Missouri in general? Like, are there general differences between those types of prairies?
Yeah, so Johnson County is a kind of an interesting intersection. So we're not quite the Flint Hills. We're not quite the more arid landscape a little west of here, and we're not quite the lush eastern tallgrass prairie. And so we're kind of in a transition zone in between. So you'll see a lot of plants that you reference from central Kansas, you'll still see here in Johnson County, but you'll also see stuff from further east into like the St. Louis area. And so we're kind of an interesting transition kind of between some prairie zones here. So we kind of get a little bit of both worlds that you see kind of further east and west. And our oaks are in transition and hybridize. And so where the state line is kind of interesting. So you'll see things like dotted blazing star that is, you don't really see it in Jerry Smith Park on the Missouri side. Or erygeniums like the Leavenworth eye, like that just starts kind of here. And so we're kind of on a tipping point, kind of between two different prairie landscapes. It's fun.
That is really fun. Okay, gosh, our audience is so engaged today because this is such an awesome topic. I'm really, really excited about this. So we're gonna go into some more questions here, but first, let's jump into our pop quiz, shall we? So we mentioned at the top of the show that we do a pop quiz to test your knowledge, see if you're paying attention during today's presentation. So I want everyone in our audience to go ahead and find the chat button, webinar chat. This is where you will put your answer. And I want you to find that blue box and click everyone because I want everyone to see everyone's answers. And the way the pop quiz is going to work is I'm going to ask the question, and the first person to answer correctly will receive these very cool, see if I can get a better shot here. Let me hold on. Let me get rid of my background real quick so that you guys can see this. Let's see if that works. Maybe a little better. Okay, so we've got two posters from Shawnee Mission Park and Big Bull Creek Park, courtesy of Johnson County Parks and Recreation District and their Heritage Foundation. So the question is this, and we'll also put the question into the chat there. So are you ready? It's a two-part question. Name two resources Arbuscular Mycorrhizal Fungus transfers to interconnected plant species. Let's see if we've stumped people this time. Name two resources Arbuscular Mycorrhizal Fungi transfers to interconnected plant species. All right, we're getting our answers in. Awesome. And I'm going to rely on your expertise, Liz, to determine our winner. It's going to be the first person who answered correctly to everybody in the chat.
Well, carbon does move, it's true, but it's the plants feeding the fungi carbon. So I'm going to have to give it to Rebecca, who said nitrogen and phosphorus. So that's what the fungi are getting from the soil and giving to the plant.
Awesome. That might have been the hardest question we've asked so far in Curious by Nature. So thanks everyone for playing, and congrats, Rebecca. Be sure to respond to the email that we're going to send out. Send us your mailing address and we will send you these awesome posters courtesy of Johnson County Parks and Johnson County Heritage Foundation, and as well as some Deep Root swag. So you'll be getting that here soon. Thanks for playing, everyone. Okay.
And Sydney, we do have, I think, a poster now for every single one of our parks by the end of this year. So we're working through the final phase of that right now. And so I think the park poster art program is about to wrap up, and we should have an iconic image for every single JCPRD park at the end of this year.
Oh, that's awesome. I love these. They're in the style. How would you describe the style, Matt?
Like the WPA kind of Great Depression era national park style posters, I guess you'd call them.
Yes, I love it. I've got one of Kill Creek Park, and it is hanging up on my office wall right in front of me. It's
Yeah,
really fantastic.
supports a lot of local artists. It's a real great opportunity for emerging artists to get something out there because it's a real low level of entry, kind of getting something commissioned. So it's a great way to support the local art community and tell the story of our parks.
Yes, it is. So you heard it here, folks. Our local artists are making these posters. They're stunning. So if you're not Rebecca and didn't win today, you can go online and get yourself one as well. And I'll be sure to include a link to that in our resources. Okay, we've got time for more questions, and boy, howdy, we got a lot of them. So we're gonna go to the lab, if you will, and we've got a great question from Christiana. Christiana asks, what is the propagation process once you have extracted your fungal spores in the lab?
Yeah, good question. It's a long one. And so I showed you on that little microscope, there were some white fungi that were big and some circular and some smaller ones that were black. So color, shape, and size are some of the traits we look at to identify these fungal species. So oftentimes the first step is to sort out these fungal spores into little piles in the microscope, which involves, very fine motor skills with my hands and not drinking very much coffee that day. So even a little bit of the shakes, it's pretty hard to do. So yeah, we make sort of piles of spores that we think are the same species. And then, as I mentioned, these fungi are obligately dependent on plants. So to grow them up, we take that spore pile and we put it in a sterile growing media. Usually it's some like sand and soil that we've autoclaved, and we put a plant in there, and then we pipette those spores right on the root. And we allow those few spores to grow with that plant for 4 to 11 months, depending on the species. Some take longer to grow and make spores. So they'll grow, they'll be fed by the plant, and they'll perforate throughout all that sterile soil matrix. And then that becomes inoculate we can use. Another major challenge with growing these fungi is that we have to grow all of our species separately, otherwise they compete with each other. So growing them in the lab is different than how they would grow in the wild, where there's lots of different plant hosts and like rocks and spatial structure they can spread out in. In the lab, they're growing, in pots in this media, and there's fewer places for these fungi to sort of differentiate from each other. So if we grow a bunch of species together, we lose diversity over time. So when I'm creating inocula, like for these Johnson County Parks projects, and I want it to have 12 species, I'm growing 12 different pots of fungi that I then combine and mix together in a thing that eventually goes out into the field. But the whole process from like going from a prairie, doing all that microscope work, and then bulking up the fungi usually takes a couple of years to do.
Wow, so very tedious, labor intensive, and that boggles my mind. I'm not surprised, but at the same time, wow, that's crazy. Yeah.
Yeah.
Very hands-on. Okay, so we're going to now shift to a question from Kathy. And this will probably be a question for you, Matt. How can you tell it's the mycorrhizae that are causing the abundance and not just the excellent land management?
Yeah, so I think that's more of a Liz question, but I will say that sometimes we will heavily overseed projects versus a bare minimum seeding. And sometimes when we do a planting with like 20 seeds per square foot due to rodents and erosion and wind. Like, so the pump house prairie was on a slope, and the lower slope there had less plants prevalent than the upper slope and things like that. So there's a lot of things at play. So we will generally try to really, if we can afford it, really overseed projects to ensure success and diversity, because you leave a lot up to chance when you go with a low seeding rate and then allow the environment and rodents and things like that to get involved. And so we try to set the prairies off on a good track record with as much seed per square foot as we can afford.
Nice.
And then I'll just add to that. So these are restorations that are experiments. And so for science, we have controls. And so, for example, at the pump house, everything had the same seeds. And then we had 3 plots that had AMF added and 3 plots that didn't. And so we kind of compare the plants that establish across those controls. And then at the Big Bull Creek Park experiment, that was even bigger landscape, so we could have more replicates. We had 5 controls at that site and 5 that had the different inoculation treatments. And so comparing across the two is one way we look at the shifts and what we think are caused by the mycorrhizae.
Nice. I mean, you're getting the data right there. I think that's really cool. And can you remind us how long have these studies at Johnson County Parks? How long have you been doing them there?
Yeah, so the seeds went in. This is their second growing season, and I had to grow the mycorrhizae a couple seasons even before that. So we've been talking about this, it feels like, for a while, Matt.
Yeah, and if you want to make an order with Liz, you really have to think forward. Like, if you want to plant a prairie, in 6 months, she's probably not going to be ready for you if you have a big project. So you've got to kind of have it, a longer thought process on how much seed you're putting out and the acreage and how much she can grow. Because her organization's not very large, her personal business, and so you really have to kind of think of how she's going to scale up for your project.
Yeah, that makes sense.
Absolutely. We
Nice.
always have time for like small, small additions, but we're kind of B2B or project to project sort of thing. So yeah, if you're doing big projects, it definitely helps to have some advanced communication so I can grow enough.
Yeah, that makes sense. Okay, man, this is tough because we've got 10 questions here, but probably only time for a couple more. So I want us to maybe quickly, Liz, is there a reference that shows which plants rely or need AMF more than others? Is there something out there?
Yeah, good question. So I have a database of prairie plants that I've grown with and without mycorrhizae in the lab to get a sense of like their absolute response to these native fungi. And so right now I think we're at like 170 grassland species, and we've done some like weedy things like Johnson grass that we don't like, and some of our late successional things that we love, and everything in between. And so, yeah, I can share that list. I don't know the best way to do that. If you would share it out with people, or you can email MycoBloom, and I can get you a PDF copy of that too.
Yeah, actually, if you want to email that to me, we'll include it in our show notes so that we can send it to everybody. That would be fantastic.
Sure.
Okay, so a couple more relevant fungus questions. I saw a couple that were really intriguing. We're going to go with Brenda's question. Brenda says, I use bokashi, a microbe anaerobic form of composting to make my home compost. And they say they do see fungal strands in the compost once it cools and is cured. They're presuming that those are not the same type of fungus, but their native plants seem to love the compost dressings. Are they creating their own ecotype of fungus? That is the question.
Yeah, great question. And compost tea can do wonders. So you can grow a lot of different microbes in compost tea, tons of bacteria, tons of fungi. And you can grow fungi that make little hyphae, but it's not possible to grow arbuscular mycorrhizal fungi in compost tea because they need a plant to feed them. They can't really make their bodies or excess carbon in any other way. So you can grow lots of microbes, including fungi, with compost tea, and that can really help your plants, but not these AMS specifically.
Gotcha. Okay. And then Carolyn is asking, do you think there will be a time when you're able to purchase the native seeds with the inoculant already added?
Good question. I don't know.
A girl can dream though, right?
Girl can dream.
So I think some of your bigger projects, have you inoculated plugs for like commercial projects?
Yeah, so usually with the bigger projects, I'll give a bunch of inocula to the nursery, and when they're growing their first upsizing or whatever, they'll put it on their seedlings, weeks before it goes into the field. So on the bigger project, that works. For these Johnson County projects, we were putting it in the large pieces of equipment that you guys have to put native seed out there, like seed drills and that kind of stuff. And yeah, people have mixed the seed together with that, like on their own. But so far, we haven't offered MycoBloom and a seed mix, as a unit. You kind of have to mix them yourself, but it's not so hard to do.
That makes sense. Okay, we've got time for one more question. Oh, this is tough. Okay, I'm gonna ask Susan's question because I feel like this is really integrates what a lot of home gardeners are doing. And I know our viewers, a lot of folks have smaller footprints. But Susan's asking, does solarization reduce fungi in the soil? And I think this is an important question, especially because there's a lot of different methods to start gardens, like solarizing, lasagna composting, or tilling. Can you talk about maybe the pros and cons of those methods as they relate to how they impact fungus?
Yeah, I mean, when you're starting a prairie, right, you have to get rid of the vegetation some way. And any way you're going to do it is probably going to harm your microbial communities. If you spray, if you solarize, if you till, those are all going to mess with your soil a little bit. That's definitely true. Yeah, so we've done some experiments in solarized areas and after herbicide, and we do see that inoculation helps after these treatments. So, we definitely think that there's some direct harm from some of these things. Solarization gets really hot. That's how it kills those plants. And so any microbes, not just mycorrhizae that are at that soil surface, are going to be cooked. Yeah.
Are any of those methods like worse than like are there ones that are better method? Because knowing that there will be some losses, right, as you mentioned, but are some methods better than others?
Well, my personal strategy, I do like solarizing because it's really good at cooking off grass, which is usually what I'm trying to kill in my yard. But I have access to a ton of microbes. So it's really easy for me to add some back in after, I mess with them a little bit. Yeah.
Okay, good to know. All right, guys. This is fantastic. Dr. Liz Koziol, Matt Garrett, thank you both for joining us for today's episode of Curious by Nature with Deep Roots. Folks who attended live, you will receive the show notes soon after today's program, which includes really great resources, links to Deep Roots' upcoming events, MycoBloom, and all the species discussed in today's show. So if you have any questions, be sure to reach out to Deep Roots. We're here to help you with your native gardening journeys and connect you with resources. So thank you all. And Liz and Matt, I hope you have a great rest of your week. So good to see you.
Thanks.
Thank you.
All right. Take care, everybody. Have a great rest of your week.